to start the last presentation, the final presentation. I'm going to be talking about variant prioritization and association. If you have any questions, you can ask them about the exercises. You can ask them on Slack, and we will also answer them at the end, during the presentation or at the end of the course. You will still have time to work on the exercises since the virtual machines and the material will be available to you for the next 2 days. So I'm going to start with this talk. I will be talking about predicting consequences of genetic variants using Ensemble, some statistical methods for variant association to the phenotype, like common variant association, rare variant association, and how combined test works. I'll talk a little bit about the guidelines for investigating causality of genetic variants in human disease, choosing filters and follow-up experiments, and I'll give a little bit of conclusions and closing remarks. So first, I would like to start the presentation with the quality control. And I cannot express how important it is to be sure that you have a high quality data before you start making any associations, because this is the most important part of your analysis. Sometimes in any project, you will spend 6 months maybe making sure that your data is good to go, making sure the quality control steps have been made, and then you can perform the analysis and the association tests in 1 week probably. So be sure that your data is high quality and that you can move to the next step. So what is predicting consequences of genetic variants? In order to make sense of the increasing amounts of sequencing data that are being produced, it would be useful to know the effect of the detected alleles on transcripts, which may aid prioritization. Doing this manually is laborious and error-prone. So we know that the DNA encodes all the information to form proteins. And this information is organized in triplets. Each triplet encodes one amino acid, one specific amino acid. So let's say you have your table here of all the combinations of bases, genetic bases, and how they build each specific amino acid. How would you find out what the consequence of a variant in a specific position, for example, chromosome 1, that it's a change from C to T in yeast, is impacting the protein? Well, in this case, to solve this, you would have to use the genome browser like Ensemble or the UCSC genome browser and search, see what proteins encoded on that region and where the variant falls on, and then try to predict the effect of the amino acid sequence from this change of bases. So you would have to look at whether this base results in a different amino acid in the protein. So there are different algorithms that can help with this. And the most common are the Ensemble Variant Effect Predictor, SNP-F, ANOVAR, and ANOVAR. And they differ in their ease of use and the species they can analyze and the data that they contain. But all of them are very useful. I'm going to talk specifically about Ensemble. What is Ensemble? It's an open source genome browser with a large amount of data available to explore and a number of biological annotations. And you can find it in this webpage, www.ensemble.org. It hosts a lot of genomic assemblies, and the way it works it has a very advanced database that performs automated gene annotation when you're looking for any gene. It also holds information on variation, small and large scale sequence variation with phenotype associations. It also has information on genome alignments and gene trees, so you can work solving problems of comparative genomics. And it also has a lot of annotations on potential promoters and enhancers that turns out into being DNA regulation. So you can also see whether your specific variant that you're looking for has an impact in all these matters. So Ensemble has the variant effect predictor, which is a tool that determines the effect of variants. What are these mutations doing biologically? What does it mean to have these specific mutations? And this is done with SNPs, with insertions, deletions, copy number variations, or structural variants. And it works in different levels. It can tell you about the affected gene, what transcript does the variant fall, and what is the protein sequence that change due to this variant. It can also tell you what's the pathogenicity of the variant, what's the frequency of the variant in the population, what are the regulatory consequences, whether it changes a transcription factor being inside, for example, if there's any splicing consequence related to the presence of this variant, if there are any literature citation that can tell you more about what is known about the variant that you're looking for. I also have a lot of filtering options to let you see different transcripts of the same protein, for example. Ensemble has, for each variant, it can give you different types of consequences. And these consequences are mapped, can fall in different regions of a specific gene. For each variant that is mapped to the reference genome, we identify with Ensemble all overlapping Ensemble transcripts. For example, we can see here in the image that we have the 5', we have 3', and this is a graphic representation of the gene. And we can see that the regions between the gene are, between this transcript are tagged differently. For example, this would be a region that's tagged as a regulatory region with transcription binding sites, transcription factor binding sites. This could be an exon, this could be an intron, and variants that fall here then would be splice donor variants or splice acceptor variants, and so on and so forth. Ensemble uses a rule-based approach to predict the effects that each allele of the variant may have on each transcript. Note that each allele of each variant may have different effect in different transcripts. So in this picture, we see one transcript and maybe one variant would fall here in an exon, but in the next transcript, the variant will actually be a splice donor. So you have to keep that in mind. And this is a classification that Ensemble uses depending on the geographic placing of the variant that you're looking for. But there are also, there are also several consequences of these variants. The terms that are shown here are in order of severity, the more severe to the less severe, as estimated by Ensemble, and this ordering is subjective to what the variant is predicted to be doing to the protein. And you should always make sure that this variant is actually doing what the predicted effect says it's doing. And take this information with your own judgment. The impact rating that we see here on the last column of the image of the table is a separate rating given for compatibility with other variant annotation tools. So this makes Ensemble more compatible with SNPBEV or ANNOVAR, which are the other tools that I already talked about. And you can see that variants here are, for example, predicted to be transcript ablation, or for example, you can tell if a specific change in the DNA can cause stop loss or start loss, and so on and so forth, to the effect that the variant has in the genome. Talking about the pathogenicity that you can find in Ensemble, Ensemble rates variants using two algorithms, SIFT and polyphen. SIFT uses two categories, tolerated and deleterious, and polyphen goes from benign to possibly damaging to probably damaging. And in order to categorize variants in these specific categories for the two algorithms, it uses a scale that goes in SIFT from 0 to 1 and in polyphen from 1 to 0. And these two algorithms differ from each other in the way that one of them uses ontology information and the other one is more about how the protein will be impacted in its 3D dimension, or how would it be impacted in if an amino acid changes, for example, from a basic to an acid amino acid, how these would impact the final protein. Another thing that we can find on Ensemble is frequency data in the population, which is very important. Ensemble will give you if a specific variant, will tell you if a specific variant is more common in the African population, American population, and so on and so forth. Like this one is an example, and we can see that this T is common in all of the populations that are reported in NOMAD. And this information, it's very useful because it is important to know how common your specific variant is in the population that you're analyzing, or in populations that are very similar to your population, to know, in order for you to know whether this variant is related or not to a specific disease. And this will come useful when you do some statistical methods, when you use some statistical methods for variant association. And in this way, we can split variant association analysis into common and rare variant analysis according to the frequency of such variants in a certain cohort. So let's look at this image that I have here in this slide. This image represents how usually variants appear that are related to a disease. Variants that are pretty common are usually implicated in common diseases. This is because if you have a variant that's common in the population, then and it's related to a disease, then you expect that disease to be also really common in the population. But you usually expect to see the majority part of your population as healthy. So it's still very rare in its allele frequency, as you can see on this table. And it's not only the only variant that's causing the effect or that's causing the disease. These variants are usually in strong linkage to equilibrium with other variants, and these ones are not the only ones that causing the disease. This is usually, the disease would be usually caused by several variants that are common in the population, and this is due to the redundant systems that we see in a cell. Sometimes when one part of the system fails, we can see other 5 systems that try to overcompensate. So this is how complex diseases work, right? We don't have only one cause, but we have several causes. And also usually the environment is important in the development of these diseases. And when we look to the other side of the graph and we see the rare alleles, these rare alleles are very rare in the population with a very low allele frequency, will be the alleles that are causing Mendelian diseases. And these are the ones that you can see more often in diseases that can be inherited from one person to another one in the same family. These rare variants are usually very rare in the general population. They're usually new mutations, and they also have weak correlation with other SNPs in terms that they are not in linkage disequilibrium with a specific haplotype. So it's very hard to identify them by genetic means, by looking at the population or by genotyping the population. And then we have something in the middle, which is there are low frequency, relatively low frequency variants that have an intermediate effect in the disease, and these variants would be also related. Like you would also need an exposure to environment, or you would also need a set of them in order to become diseased or to actually find an association with this variant to the disease. The way this association to disease is analyzed for the rare variants, the very rare variants, are in families analysis. You have to look into the families and how a specific disease is inherited within the family. Because they are too rare, you usually have to use next generation sequencing to identify them. And for the common variants that are really common on your population, you can just genotype a cohort sample of the population, and you can look at the phenotype and how it is segregated along with the variant in the population that you're analyzing. So the principle of an association testing is something like this table. I like to give this example because it's pretty obvious. We usually won't find this commonly. This is a very highly common marker that I'm using here in the example. And this is not what we usually find in reality, but I made it like this just for educational purposes. So let's say you have two markers, marker 1 and marker 2. In a population that's composed by 100 individuals, that's you have 20 cases and 80 controls, for example. And this is how you can see the marker 1 would be in only 18 cases, marker 2 would be in 15 cases, 15 of the cases. And then in controls, you would see marker 1 in 72 of the controls and marker 2 in 4 of the controls. So we can see that overall, marker 1 shows up in 90% of the general population, right? Both in cases than in controls. So it's harder to say that marker 1 is related to the disease. In the other side, marker 2 shows up in 19% of the general population, which is very rare. Well, it's rare, relatively rare in the general population, but we can see that it's being shown in 75% of our cases. And only 5% of our controls. So in this way, we can see that marker 2 is way more common in the cases. And that's how we can relate marker 2 to this specific disease that we're looking at. And there are several tools that can help you do this statistical analysis. And the favorite one, for me at least, is PLINK. PLINK is a free open source whole genome association analysis toolset that's designed to perform a range of basic large-scale analysis in a computationally efficient manner. And what PLINK does is it has a lot of tools inside. It can help you filter and report for standard metrics that are useful when you're doing these genotype-phenotype associations, like genotype rates, allele frequencies, the Hardy-Weinberg equilibrium to see whether your variants are being represented as you expect in the population. It can also help you detect Mendel errors. PLINK has also tools to test for non-random missingness, individual heterozygosity estimates. It can also help you check or impute sex based on X chromosome, which is helpful to know to guarantee that your samples have not been contaminated, and can also help you with SNP pruning due to the linkage disequilibrium. And this is an interesting tool that can help you detect independent SNPs that are not inherited within an haplotype. For the association test, which is what we're most interested in, PLINK can perform a case control analysis, case control associations, can also perform family-based association tests, quantitative traits association and interaction tests, and it can also detect association conditioning on one or more SNPs or another covariate like age or like population stratification, etc. The way you usually use PLINK, the way PLINK works is that you would call the tool, typing PLINK in the terminal, and then you would say what type of file are you using. You can use BCA file. You can use also PLINK has its own file formats. And then you name the file or you give the address to the file. And then you ask for an output and you name the output. And this is pretty easy, pretty straightforward. In the middle between the file, between your input file, you can add some flags on what do you want PLINK to do. If you want PLINK to make an association test or a Hardy-Weinberg equilibrium test, etc. Why PLINK is the favorite tool or very useful tool is because it doesn't alter data in place. Every time you get an input data, it won't change that input, so you don't need many copies of the same file. It also requires explicit instructions, which can be a pain for people that's starting to use the tool, but when you get to know the tool, it becomes a very strong skill from it that you must be very explicit on what PLINK should do, and that way it makes you more certain about your results. And it also produces detailed logs. So every time you run an analysis, PLINK will produce an output, and it will also produce a log of the thing that was performed on the input. And this log file would have what was the input, on what date it was performed, what was the thing that was performed to it, and how many samples you have, how many samples were analyzed. So it's also very helpful because when you go back in time and you want to see what you did in order to obtain your results, you can also rely on the logs from PLINK, and you can look at what was your input, what was your output, and what was performed to get to your conclusion. So that's also very useful. So let's say you have a file called data, and then let's, sorry, there's a mistake here. Data should be with the lowercase d. So let's say you have data with lowercase d, and that's your input, right? And you want to perform a Hardy-Weinberg equilibrium test or get the Hardy-Weinberg information from data. So PLINK would give you as an output data.Hardy-Weinberg equilibrium. And that way you can distinguish between both files. And then let's say you want to filter the data into whatever happens to pass the Hardy-Weinberg equilibrium test that you just performed. PLINK would give you as an output a new file that's called data.filtered. And then let's say you want to make an association test in this filtered data, PLINK will give you an output that's called data.filter.association. And in this way, you can also know that this final file with the association results is not performed on the original data, but it's performed on the filtered one. So that makes it very easy to work with and to work with your files. It becomes your files more organized. So this is an example on how to use PLINK. You usually just type PLINK, call for the tool, and then I would say I'm giving a BCF as an input, so minus BCF, give it an input, and then dash a sock, which is a test I want to perform, and then I want PLINK to give new names to my variants. So I would say just set missing varieties, and I would say that I want the name of my variants to be first chromosome, then position, then this symbol would be for chromosome, this symbol would be for position, and I want them to be separated by a semicolon, by a column, sorry, and then give me the reference allele, which is the first column of the alleles, and then give me the alternative allele. And that way I can assign a specific tag to the IDs, to the variants that don't have already an ID. And then I would give PLINK, because I'm asking for an association test, I would give PLINK phenotype information on my cohort. So I would just say, well, use this flag to say, here's a phenophile, and then specify what would I like to be in my output file. In this way, in this example that I'm showing you, PLINK will perform an allele-based test and will output the odds ratio for each one of the alleles and how they're associated between cases and controls. So what's the odds ratio? The odds ratio is the ratio of odds of an event in one group versus the odds of the event in the other group. So let's say the event here is having allele 1, or the other events having not having allele 1, right? So it's related to the relative risk of having to be in one group if you are exposed to the event. But it's not actually the relative risk. The odds does not represent the true risk. Its value is close to risk when the event rates are low. And when we're talking about genetic variation, the event rates are always low. So the relative risk is a ratio of risk of an event in one group. An example, this is the exposed groups of people that have the mutation versus the risk of the event in the other group. I mean, the group that is not exposed to having this mutation. If the odds ratio is bigger than 1, then the allele that we're studying is at higher frequency in cases than in controls. And then we can conclude that it is a risk allele. And so forth. If the risk allele would be the mutation, then we can say that having the reference allele, not the alternative one, but having the reference allele is a protective allele, or it would be protective to this condition. So I already said that most variants that are associated to disease are rare, at least for 2 rare diseases. So studying rare variants is very hard. There are many more rare variants than common variants in general population. And the disease-associated variants are expected to be rare because you would like to have your population, you would expect to see your whole population being healthy, right? I mean, the most common phenotype in your population should be healthy. So to detect them, as I already said, sequencing is needed. And well, in order to analyze the association between these rare variants and the disease, you have to perform these rare variant studies that are usually seriously underpowered. So let me give you an example of this. Let's say you have a cohort of cases and a cohort of controls, right? And let's say you have this variant here. That's the one that is inside the square. And you see that this variant is more common in cases than it is in controls, because you can see that there are 3 cases that carry it and no controls have it. In order to know if this is true, even if you have 10,000 cases and 10,000 controls, the minimum p-value that you're going to get of a 3 to 0 observation, which is what we're looking at, is 0.25. This means that it won't even reach nominal significance. And this makes it very difficult to associate these rare variants to a disease. So in order to overcome this, scientists have come with aggregation analysis of rare variants. And these aggregation analyses are single variant because single variant test is not powerful enough to identify these rare variant associations. So what we do when we do an aggregation analysis is try to test the joint effect of rare variants or rare and common variants while adjusting for some other covariates. There are different methods to do this: the burden or collapsing test, supervised adaptive burden or adaptive collapsing test, variance component tests that are also called similarity test, and omnibus test. The burden test, how they work is that they aggregate rare variant information into a summary dose variable. These type of tests are more powerful if all rare variants are causal variants with the same effect sizes and association directions. And there are different algorithms like binary collapsing, count collapsing, and weight sum tests, but they all work in this manner. Let me explain. So let's say you have your cases and your controls. Y is a phenotype, right? So it's 1 for cases and it's 0 for controls. And let's say you have a group of variants in a specific region or a specific gene, and you can see that if you analyze these variants in an individual way, if you calculate the risk that's increased because of having these variants, you would say that, well, having this variant make not an actual difference between cases and controls, right? And you go to this one and you can conclude the same thing. Having this variant is not very hard. It's not an actual risk to become diseased. If you look at these 2, for example, you would say individually, you would test this one and you would say the same thing, but what happens if you join them? If you collapse them in a summary dose variable, which is that you count all the variants are in this region, and then you can see that this specific region has a heavier load, also called burden of mutations. And then you can say, well, maybe not one variant, but a set of variants, or having 1 or more variants in this specific region is more related to being diseased, to be part of the cases than to be part of the controls. So this is a very interesting way to analyze this rare variant association. But there are other ways, like the variance component test. And these variance component tests address the burden test limitations on different effect directions and many non-causal variants. So if we go back to the burden test, if we were to put maybe 1 variant here, for example, we would see that having two variants in the same region for the cases would increase the risk, but it would not be significant because then you would have one protective variant. But at last, it's not one variant that's causing the disease, it's the two of them, right? At least in your population that's causing the disease. So the variance component test addressed this problem. And by modeling distributions or modeling different descriptive statistics from cases and controls in any specific region. And there are several other algorithms that do work with these variance component tests, like the C-alpha test, the sequence kernel association test, and the omnibus test, and the variable threshold test. And I kindly invite you to read this review that has a lot of information. And here's a comparative table on how burden, adaptive burden, variant component tests, combined tests that combine the effect, combined burden test and variance component tests are compared to each other. What are the advantages and what are the disadvantages? And you should be able to know which one is the most appropriate to your project, to your specific question. And I also would like to recommend further reading. So if you're interested in doing some rare variant analysis or rare variant association, you can have a much more wider, much more information about how it's done and what are the methods that you can use. So about the guidelines for investigating causality of sequence variants in human disease, there's a beautiful paper published by MacArthur that's titled like this, and they published several recommendations, and I put some of them here. So first off, provide complete positive and negative evidence associated with the association. Try not to be biased, even though you think that your variant is strongly associated to the disease. Try to look also for the other side of the story. Whenever possible, place genetic information and experimental results within a quantitative framework. What is the probability of observing this result by chance in a random selected variant or gene? And this is very important because we are prone to bias. So when you try to make it more quantitative, your experiments and your analysis, then you can control on that. Look at public data sets of genomic variation, functional genetic data, and model organism phenotypes, so you can confirm that what you're observing is also repeated in these model organisms, for example. Reassess previous reports of gene or variant implications and rigorously as your own data, not because it's published, accept that it's real. And describe and assess clearly the available evidence supporting prior reports of gene and variant implication. So here's an example of why these guidelines are very important. This paper was published in 2016, and what this paper said in their highlights was that the authors found an association between a specific variant, a specific mutation in this gene, NR1H3. It's a mutation that goes from an arginine to a glutamine in, I think, position 415 in the protein. And they found that this mutation results in loss of function of the gene, leading to transcriptional dysregulation. And they linked it to familial multiple sclerosis. Later that same year, 2 papers were published. In the first paper of them, the authors proved that this variant is very frequent in the population. And turns out that multiple sclerosis is not as frequent as this variant, so it should not be related to the phenotype as the other paper said. So one of the highlights that they report is that there is no evidence of variant enrichment in cases compared to population controls, since it is pretty common in the population. And in the other paper, they increased the number of subjects that were studied, and they saw that in a 13-fold larger sample, they found no evidence in association to either multiple sclerosis or clinical course. So these both papers helped us conclude that the result of the first paper is a false positive due to insufficient statistical rigor and flawed logic. And this is with the use of a bigger cohort or with the use of publicly available data on how these variants behaved in the population. So continuing on the guidelines for investigating causality, if you have a gene that you can say, well, this gene is really associated to the disease, then be sure to, if it's a presumed monogenic disease, evaluate the genes previously implicated in similar phenotypes before exploring potential new genes. Also, report a new gene as confidentially implicated only when variants in the same gene and similar clinical presentations have been confidently implicated in multiple unrelated individuals. And at last, in all cases possible, apply statistical methods to compare the distribution of variants in patients with large matched control cohorts or well-calibrated null models. This will help you be sure that what you're seeing is not happening by chance, but it's because it is actually related to the phenotype or the disease that you're studying. If you have candidate pathogenic variants, determine and report the formal statistic evidence for segregation or association of each variant and its frequency in large cohort populations match as closely as possible to patients in terms of ancestry. Recognize that strong evidence that a variant is deleterious and or damaging is not sufficient to implicate that the variant plays a causal role in the disease. Predict variant deleteriousness with different methods, but be sure that these methods are not independent, not necessarily independent lines of evidence. Validate experimentally the predicted consequence. If you think that this candidate variant is going to kill the protein, then make an experiment to prove it. You can do this using assays or patient-derived tissues or well-established cells, cell lines or animal models, and avoid assuming that implicated variants are fully penetrant or completely explanatory. As I said, the cell is filled with compensatory mechanisms that can fix for one specific error, for most of the errors. Now, thinking about variant filtering for finding disease-associated variants. So this is the interesting question here is, how do I choose the filters and follow experiments to find these disease-associated variants in my genomic data? First, you have to think a lot about your project needs and your project characteristics. And this not only is about the quality control, but how you design the experiment. If you're studying families, for example, and you have DNA from different members within the same pedigree, perhaps it is a good idea to use more permissive quality filters. I mean, at last, you can ensure that no variants are lost when applying filters if you are more permissive. Co-segregation and resequencing by an orthogonal method, would ensure the true positives. If you're studying a rare disease in family setting, large disruptive changes can be expected from the causal variant. So you can assess these with some follow-up experiments on functional cell line or tissue assays, as well as checking phenotype data from consortiums such as the Knockout Mouse Phenotype Program or the Zebrafish Information Network. And in this way, you can assess whether the specific variant that you're looking at has already been reported to change the phenotype on these model animals. On the other side, if you're studying large case control cohorts and do not have access to the participant DNA, you need to be more restrictive in your filters to increase the specificity. And maybe you would have to ask for the GVCFs to correctly assign non-carrier status to these specific variants. These projects usually implicate generic variants of a small effect because you're working with bigger populations. So bear in mind, these may be common in the population, and phenotypic information from the model animals may not be informative. In this case, maybe a large number of individuals would be necessary, so the associated phenotypes can be checked in large genotype-phenotype databases like the UK Biobank, for example. So my conclusions and closing remarks. Finding disease-associated variants for next-generation sequencing data is not trivial, it's not very easy, but there are a number of recommendations we can follow to increase our chances. First, software such as Ensemble can help us collate information about predicted protein consequences, potential pathogenicity and allelic frequency in the population, but we should always evaluate the full extent of positive and negative evidence of association, including performing statistical evaluations of finding a result by chance and reviewing available data on association phenotypes. We should apply the most appropriate filters to our data depending on our experimental design. And here I can also add that it's very important, well, as I already said, to check on publicly available data and to plan and follow up experiments so you can guarantee that the association that you're looking at has an actual biological explanation for it. Well, you need both. You need the statistical and the biological explanation for it in order to prove that it's true. So with this, we conclude our workshop. Thank you so much for your attention. Please don't forget to complete our post-workshop survey. You can find the link in the Google Classroom. As I already said, the virtual machines, the instances, and all the material of the course will be available for you for the next 2 days. So if you want to complete the exercises, you can do so. And please ask us if you have any questions through the Slack. And that's all for now. Thank you, everyone.